So good morning, everyone. I'm going to start in the next minute or so. So if we can take our seats, we can get the day going. We have half a day dedicated of the 4-day marathon meeting. Thank you very much for your time, effort, and for being here, and especially being here this early in the morning on a 4-day meeting. We have only half a day to get through the rest of the agenda, but we should be able to do it if we keep on time, which is what I'm going to try to do right from the start. So today, I'm going to spend maybe about 5 or 10 minutes just giving a little bit of some introductory remarks. And welcoming and opening up the session, which Angela would lead right after it on surveillance and data that's been used to inform, record, and influence not only clinical practice but also potentially clinical trial planning. We'll have a break shortly after that, and then reconvene with the ReSec-TB platform. So this is sort of a bookend. We've started the RDST consortium workshop within CPTR with ReSec-TB, and we sort of want to end it on that note as well. And then we'll have a closing address, and I'm really excited to have Tiong Zung Ting from Malaysia, which is our patient advocate this year, and I'm sure that we'll find that extremely interesting. She's a very engaging speaker. So why don't we have... Yes, my first slide. So I thought I'd just give a brief overview. Again, I know that there's not a lot that I can say that hasn't already been said over the past 3 days, but just to remind us of the progress that has been made over the past 15 to 30 years with regards to the number of lives saved, the decline in TB mortality reaching almost 50%. The amount of progress really quite dramatically, because it was started quite late, on the HIV-related TB deaths, which has been reduced by about a third over the past decade. And despite the effect that we have on MDR-TB and the crisis of MDR-TB, the one piece of information I thought was really a good piece of information was that the treatment of all the individuals that were actually identified, nearly all of them were put on treatment, which is an amazing statistic when we start taking a look at the cascade effect of other diseases. So I thought that was worthwhile mentioning. But of course, we do still have some challenges. Of course, there's a funding gap. But of the 9.6 million people that we know that are infected with TB, at least that's our estimate, we also estimate that 3.6 million are not. So that equates to approximately 62% of individuals that we've identified as being TB infected, whereas we're missing quite a few. Despite the progress on the HIV-TB response, we still need to accelerate it. There's still a lot more work to do, even though we have had some success over the past few years. And of course, I'll be remiss without mentioning MDR-TB and XDR-TB. And the issue that we have now is the identification of those individuals, because if we can't even identify them, then there's no way that we can eventually hope to have those cured. And then, of course, the treatment being only 50% effective, that's another problem that, of course, the drug discovery pipeline hopefully will try to address and as we advance it to new drug regimens. So we've seen this slide before. This is that cartoon of the construct of CPTR, just to give you an overview of the different, and the Rapid DST Consortium is one arm of CPTR, which came into existence about 3-4 years ago. And just at the bottom, this is really the mission statement of what we have, is to accelerate the development of a clinically useful, regulatory approvable in vitro diagnostic assay for drug susceptibility testing. So we're being very specific here in order to facilitate the rational use as well as the deployment. So we hope to, at some point, not only have drugs available, but as we deploy them, we have to know where to deploy them and how to deploy them. And to do that, we have to have a tool. And that's what we need to do for the Rapid DST Consortium. Of course, there are 3 work groups, if you don't know. We've talked a little bit about the enabling science yesterday, and that's really to try to look at the focus on the discovery and validation of a molecular basis of drug resistance. And then we have the surveillance work group, which we'll talk about shortly in another few minutes, that Angela will be leading. And that's really to define the magnitude and the trends of drug resistance, particularly in high-burden countries, and particularly in those countries which we have very difficult amount of evidence or difficult level of evidence to point to. So we're looking for novel and new methods to do that. And of course, the community. We've been extremely lucky to have such a vibrant community to provide that input, that collaboration, in order to nucleate around separate issues. And that's really where the consortium actually started, is around the nucleation of pyrazinamide and the issues that we were having, and that continues to be the case. And as Jamie had said, it's a never-ending experiment. So ReSec-TB, hopefully, will be the hub of what this is. So we have various different touch points, not only within the community, which we hope to be able to do, but also within the various work groups, and hopefully then to lead into the information required to help the advancements of new diagnostics. So ReSec-TB, although within the RDST consortium, has really, the leadership has come up from essentially 6 different organizations, each one of them providing their own input, which has been a key in this, is that we're moving forward as a whole group. And that not only includes the Critical Path Institute, but also FIND, the World Health Organization, the Stop TDB Partnerships, at least the New Diagnostics Working Group, and the CDC Division of Tuberculosis Elimination, with assistance and interest also of the National Institutes of Allergy and Infectious Disease, both DMID as well as Division of AIDS. So what's the vision for RDST in the future? And of course, we've now launched ReSec-TB. Later on this year, we hope to launch that now for the rest of the TB community. At this point, we're going through a number of steps to ensure that we have a viable and useful product, and that's going to take some time to do that. But during this time, we've noticed some areas of improvement, and we'll talk about these over the next few hours. One of them is standard development, and during the talks on surveillance, one of those talks is going to be on standards of the end of the reporting part of ReSec-TB. So what's coming out of it? We need to be able to standardize the language that we're using so that everybody understands what needs to happen after that. But also the standards for the input side of things. And that's, I think, where a little bit more work can be done. Of course, policy becomes quite important. And I think over the next year, I would like to start having some additional discussions with not only stringent regulatory authorities, but also the WHO in order to align ourselves with the policy. Because without policy, we're going to have a hard time on a number of different fronts. It's going to help drive global data contributions for sure, but it's also going to, on the output side of things, the use case scenarios. And we've touched on the requirements for research and surveillance, but particularly important is going to be the clinical application and for diagnostics as well. And there are a number of different areas within diagnostics that could apply in. We had that discussion with the developers on stage that Madhu had listed. And I just thought I'd take just a little bit of time to talk about that diagnostic development pathway. So CPTR really deals more along the front part of this, is the pre-competitive space, in order to identify the molecular mechanisms for drug resistance. That's the enabling science, the surveillance. The assay development work group worked closely with FIND to develop, and also MSF to develop target product profiles with the help of WHO, and then defining those use case scenarios a little bit further. Of course, there's a lot of other inputs that need to come into this, the need and necessity of samples, what Madhu has done in the past and continues to do with regards to market size, the variability of DST, the variety of different drug regimens that are being put in clinical trials, and what that has in mind, and of course policy. So eventually there'll be a point where a company will say, well, we have enough basic science and enough data to actually have an invention and include patents. But then there's a gap. they sort of hit a little bit of a wall where they have to now define those target markets and develop a better business plan in order to move forward. This is sort of a little bit what Jim was talking about as well. And this is what typically I've always, and a number of other people have said, is the valley of death. And it's a valley of death for a number of different reasons, and I'll get to that in the next 30 seconds or so. So who actually funds this part of it? And it's government funding, a lot of SBIRs, which is really great to get through that initial period after proof of concept. Angel investors, a big contributor to this if they're available. Philanthropic organizations, and they usually end up funding sort of that early stage technology development to take it from proof of concept to early stages. But then, you know, that money only goes a certain way, and venture capital is usually the group that provides funding for this. Unfortunately, because we're dealing with a disease of poverty, there's not a lot of venture capital out there to do that. So there are other organizations, international organizations such as UNITAID and USAID, but they're more involved in the back end, sort of the delivery and the implementation of diagnostics, and that's more in production and marketing. So we have this sort of stage of development from early all the way to production and marketing, and typically if you have funding, it could take, depending on the technology, 3 to 5 years. However, because there isn't a good funding mechanism in place, that often, as you've seen with CONI, can take longer. So there's a lot of promise in the beginning saying, yes, we can take another 2 years, things will be out, we have a plan, this is how it's going to move forward, and then there's a bit of funding problem. So they've got to take a step back, apply for funding. Things then end up being much longer. And so oftentimes what we see in these talks, and these, especially for small companies, is that we have a development plan, a timeline, and the timeline keeps pushed further and further to the left. There are a number of different go, no-go positions and points in this. There's you know, designing prototypes, making sure that they work, finding out what you're doing, identifying your suppliers, where they're coming from, making sure that that has quality associated with it. You eventually get to a place where you can start locking down the design, you validate your business plan that you've initially started with, you start looking at performance of the assay, you then, hopefully if that goes, then you go into clinical trials. Of course, this costs money, and in these different stages, you have to plan for it. Then eventually you get into manufacturing, scale-up, distribution, and at some point you'll hopefully have a viable business plan. But during this time, you're always operating in the red. There's nothing that's coming in. Everything is going into it, but there's nothing coming in from it. And it takes several years before you actually start realizing some return on that investment. And of course, this is all, the umbrella to this is all under policy and regulation, which is why I want to try to see if there are ways in which we can help diagnostic companies reduce that time by looking at some of these other issues. So I thought I'd stop here. I'm going to welcome Angela to the podium to begin the surveillance session. And I thought I'd just leave this one slide that actually Angela and I, a few months ago, were at Greenwich, and this is a slide I thought that she thought also was good on standards, and this is at Greenwich Village where you see the standard of time as well as length. Thanks very much.